Thank you for attending this talk. I'm Zeyang Shen, a forthcoming fifth-year PhD student at the University of California, San Diego. In this talk, I will present my preceding paper on my method MAGGIE that leverages genetic variation to identify DNA sequence motifs, mediating transcription factor binding and function. Let me first give you a brief introduction of the biological problem here. Even though cells in our body share the same copy of DNA, different parts of DNA are activated in different cells, and that gives the variety of cell types and different cell states. Transcription factors play an important role in making this possible. Transcription factors are proteins that bind to DNA by recognizing specific DNA sequences, also called motifs. In this illustration, two transcription factors bind to their respective motifs. Together, they recruit co-activators and activate gene transcription. The very important question to understand this process is what transcription factors are involved in different scenarios. The answer is usually not straightforward, considering there are already thousands of transcription factors with known motifs. To find functional transcription factors, a common strategy is to look for enriched motifs. In this toy example, a motif enrichment algorithm will identify sequence GGAA as enriched in input sequences compared to background sequences. Dozens of motif enrichment tools are currently available, but an issue we often see in these tools is that they tend to output a long list of enriched motifs. In some biological scenarios, enriched motif does not equal to functional motif. The motif might be present, but only be bound by transcription factor in a different cell type or under a different condition. In order to focus on functional motifs and potentially reduce false positives among enriched motifs, I'm proposing a new method called MAGGIE. MAGGIE is based on a hypothesis that if the change of a motif is frequently associated with the change of a sequence function, then there is a high chance for this motif to be functional. To look into this type of association, we require extra information provided by genetic variation. Suppose we have a bunch of sequences with a certain motif, as pointed out here and colored in red. These sequences are labeled with some epigenomic feature such as transcription factor binding, open chromatin, or histone modification. In order to find out whether this red motif is functional, we leverage the sequence and the measurement of the same epigenomic feature from another genome. That can be another human individual or a different allele of the same individual. We then look for the sites where this motif is mutated by genetic variation between two genomes. MAGGIE is able to test the association between motif mutation and the change in epigenomic feature. Here, we define sequences associated with the presence or a higher level of the epigenomic feature as positive sequences, and the ones with the lower level of the epigenomic feature as negative sequences. Here is a more detailed overview of MAGGIE. The inputs of MAGGIE require to be pairs of sequences from the same regions of different genomes. The first step is to compute the best motif scores for every input sequence based on 1,000 position weight matrices from Jasper database. The second major step is to compute the score difference within each sequence pair. Take the red diamond shaped motif as an example. The score difference is computed by subtracting the motif score of the negative sequence from the positive sequence. It's worth noting that the background sequence is set as uniform here, so it gets cancelled out after subtraction. By doing that, the score difference becomes the log-fold change of the binding likelihood, and it very well reflects the change of transcription factor binding activity measured by ChIP-seq reads. In this example, on transcription factor Pu1, we saw a correlation as high as 0.71 between Pu1 motif score difference and the change of Pu1 binding activity. While the correlation is weaker if we instead use a non-uniform background frequency or restrict the motifs to be at the same position instead of taking the best motif scores. After getting the score differences for each motif, the final step is to test for the bias in motif mutations. The null hypothesis is that if the motif is not functional, the mutations of it will be random, and the score differences will tend to have a median value close to 0. However, if the motif is mutated in a certain direction, it is likely that the motif is functional. Using our package available on GitHub, the significant motifs will be stored in a text file and be displayed in an HTML file for better visualization. Perhaps a toy example could help you better understand how the data flows in MAGGIE. Suppose we start with epigenomic data from 2 different individuals for a type of histomodification. After looking at their data, we found 3 regions where 2 individuals show different levels of histomodification. We believe such difference is largely due to the local genetic variation between their genomes, so we input the sequences of these regions into MAGGIE to see what it can tell us. MAGGIE will look at the first region and put the sequence associated with the higher level of histomodification to the positive set, and the other sequence to the negative set. Then it will compute the best motif score and score difference for every known motif. The illustration here is only for one motif. It will repeat this process for every region. Different from the first region, for the second region, the sequence from individual number 1 is put into the positive set, and the sequence from individual number 2 is put into the negative set. However, the score difference is always computed by subtracting negative sequence from positive sequence. After iterating through all the regions, the final set of motif score differences will be tested by the Wilcoxon signed rank test against medium 0. Motifs with score differences significantly deviated from medium 0 are predicted to be functional by MAGGIE. Starting from the next slide, I will show you how the similarly straightforward framework performs surprisingly well on different datasets. I first tested MAGGIE on simulated data. I randomly generate sequences with 2 motifs, and these are my positive sequences. I then simulated genetic variation at one of the motifs. In this illustration, the green motif on the right, and these sequences become my negative set. Here shows the results from MAGGIE on 3 different motif pairs that were randomly selected. In terms of prioritizing the functional motifs, MAGGIE outperforms other motif analysis methods in all experiments. For the sake of time, I will not get into details on this, since I would like to save time for more interesting biological problems. But if you are interested, there are more details in the paper. Now let's take a look at how MAGGIE performs on real biological problems. Easiest thing first. We asked MAGGIE to identify corresponding motif for transcription factor binding. I downloaded the predetermined allele-specific binding sites from literature for 2 cell lines and over 10 transcription factors. Those sites were found to be heterozygous and have difference in CHIP6 signal. The alleles associated with a higher binding activity are my positive set, and those with a lower activity are my negative set. The results from MAGGIE are shown on the right. The datasets are on the y-axis with labels of its cell line and transcription factor. The motifs are on the x-axis arranged in the same order as the transcription factors. You can see the highest significance value for each transcription factor lies on the diagonal, indicating that MAGGIE successfully identified the corresponding motif for each transcription factor. We also know that many transcription factors tend to bind close to each other, so we further tested MAGGIE to see if it can recover collaborative binding between transcription factors. We leveraged the CHIP6 data from 2 genetically different mouse strains for 3 lineage determining transcription factors of macrophages, PU1, CBP-beta, and ATF3. We looked for regions that have different binding between the two mouse strings and input those sites into MAGGIE. Similar to what we saw in those allele-specific binding sites, MAGGIE assigned high significance to the corresponding motif for each transcription factor. But besides that, MAGGIE also discovered a high dependency of CBP-beta on P1 or SBI1 motif, while P1 binding is not significantly dependent on CBP motif. This result aligns with previous findings that P1 acts like a pioneer factor to open chromatin and then recruit other transcription factors like CBP. If we look at the results from other motif analysis tools, we see that they are unable to recover the subtle difference. For example, we used Homer to identify enriched motifs at those transcription factor binding sites, and all of these motifs were given extremely high significance, which explains why you sometimes see a long list of enriched motifs from this type of tools. Another major advantage of MAGGIE in my mind is that it can be applied to all sorts of epigenomic features beyond transcription factor binding sites. In this experiment, I leveraged thousands of chromatin QTLs predetermined in lymphoblastoid cell line, which is similar to B cells. These QTLs can test MAGGIE on different features, including DNase, H3K27 acylation, H3K4 monomethylation, and trimethylation. MAGGIE identified SBI1 and other ETS motifs as the top functional motifs for all the testing features, which is consistent with the important function of ETS factors in B cells. Interestingly, CTCF is identified as only functional for DNase or chromatin accessibility and not functional for histone modifications. This suggests that the major role of CTCF is to maintain chromatin structures instead of activating chromatin states. The last and probably the most exciting application of MAGGIE I'd like to introduce here is on the stimulus response of enhancers. I first leveraged the data from 5 genetically diverse mouse strains. Those are H3K27 acylation ChIP-seq data for both basal and KLA-treated macrophages. By monitoring the ChIP-seq signal from basal to KLA-treated state, I identified activated and repressed elements for enhancer regions that have an increase or decrease in the acylation level respectively. Then I did pairwise comparisons between mouse strains and looked for regions that are activated or repressed only in one strain but not in the other. I asked MAGGIE based on those differential sites what motifs are likely functional for the KLA response, and this is what MAGGIE told us. Besides SBI1, CBP, and FOS-Jun motifs that are linked to well-established transcription factors for macrophages, MAGGIE also identified REL-A or P65 motif as functional for enhancer activation, and NF-kB1 or P50 motif as functional for enhancer repression. Both P65 and P50 belong to the same transcription factor family called NF-kappa B. So it's interesting to see how these two transcription factors can potentially induce very different functions. Moreover, this is uniquely found by MAGGIE and not by any existing motif analysis tools. To verify this prediction from MAGGIE, my collaborator Martin Hoeksma conducted ChIP-seq experiments to measure the genome-wide binding sites of P65 and P50 in KLA-treated macrophages. We saw that the P65 motif as shown here is linked with the binding of both P65 and P50, while the P50 motif is linked with the binding of P50 alone. That's reflected by a shift of the blue curve to the left where the ChIP-seq signal is higher in P50. There are definitely other NF-kappa B family members that we did not consider here, but taking what we saw here as a general hypothesis, we found that activated enhancers used in our MAGGIE analysis are indeed more overlapped with co-binding sites, while the repressed enhancers are more bound by only P50. We also saw a general increase in H3K27 acylation at co-binding sites and a general decrease at P50 only sites. All of these results validated the divergent functions of P65 and P50 motifs predicted by MAGGIE, and we're glad to see a case where MAGGIE can be used to discover novel functions of DNA sequence motifs. With that, I'd like to re-emphasize 3 take-home messages from our studies. First of all, motif score difference is a powerful metric that can reflect the change of transcription factor binding and can be used to test for association with an epigenomic feature. Our method MAGGIE was built based upon this hypothesis and is able to identify functional motifs for various epigenomic features, including transcription factor binding sites, chromatin accessibility, histone modifications, or even the stimulus response of enhancers. Finally, using the example of NF-kappa B factors in the KLA-treated macrophages, we showed that such analysis is promising to discover novel biological findings that could not be captured by other tools. We also proudly provide a free software package with dedicated documentation on GitHub. We would like to think of this work as an opening to tool development on functional motifs. We also intend to keep improving our current method and will welcome any contributions or collaborations. In the end, I would like to thank my supervisors and my collaborators for the continuous support on this project. I especially appreciate my advisor Chris Glass and my lab mates for creating such an open and collaborative research environment so that I can implement this research idea of my own. And finally, thank you for your attention.